So certainly, the evolution of eukaryotic cells was a very significant adaptation that led to subsequent rapid diversification. Another significant event in the history of eukaryotic evolution occurred after the evolution of multicellularity. This event occurred in the lineages that gave rise to algae, plants, fungi, and animals. Certainly, multicellularity allowed organisms to grow much larger and also allowed them to carry out more specialized functions. The earliest and simplest precursors to multicellular species were called colonies. So these colonies are collections of cells that are either connected to one another or move together as a group, but show very little or no cellular differentiation. Differentiation is the specialization of cells to carry out different functions. So if each cell is identical to one another, and the cells are simply working together, or moving around as a unit, then there's no cellular differentiation. Multicellular colonies can take on a variety of different shapes, including filaments or long strands, balls or spheres, or flat sheets. An interesting organism that we'll talk about later in the chapter is a slime mold. The cells of the slime mold are not actually connected, but they're amoeba-like and they move around together, giving a collection of cells the appearance of a single organism, when in fact these cells can be separated and live independently and still survive. But typically, colonial cells are attached to one another. Sometimes the cell walls of different cells have fused to one another. And in other species, proteins on the outside of the cell surface connect or join adjacent cells. Scientists are certain now that multicellular organisms evolved many times over the course of evolutionary history. Both genetic and morphological evidence supports the theory that multicellular organisms arose in unrelated lineages, independently from single-celled ancestors. So let's look at this phylogenetic tree that shows some of the major clades of eukaryotes in which multicellularity evolved. Now, there are other branches of the evolutionary tree that include other eukaryotes that are not shown here. And in at least one of those lineages, the brown algae, which include the largest protist, the kelp, multicellularity evolved a separate time. So we can see that multicellularity evolved in animals separate from other lineages because their closest relative, the chonoflagellates, are not strictly multicellular. Multicellularity evolved at least once within the fungi. Since some species of fungi are multicellular and others are not, we would have to look more closely at a phylogeny of the fungi to figure out whether this mixture of unicellular and multicellular forms represents a single event or multiple events in the evolution of multicellularity. Again, we see the red algae the green algae, and the charophytes are groups that contain both unicellular and multicellular forms. So most likely, multicellularity evolves separately in each of these groups, and possibly more than once. We see that all land plants are multicellular, representing a separate event. So let's look at one of the simplest multicellular organisms to examine the mechanisms involved in this important evolutionary event. Volvox is a type of multicellular green algae that forms a monophyletic group along with the single-celled algae, Chlamydomonas, and several colonial species. Okay, remember that colonial species are intermediate between true multicellular organisms and true single-celled organisms. Now, the Volvox is a species that was once considered to be colonial. But as scientists have done more study on the Volvox, they've found cellular specialization. And now many scientists classify it as a multicellular organism. For example, there are certain cells in the Volvox that are involved primarily in reproduction, whereas other cells do not have a reproductive function and are involved primarily in movement and metabolism. There's also a directionality to a Volvox. Even though it looks like a sphere, cells at one end have more photosensitive pigments than the cells at the other end. These photosensitive pigments allow the organism to detect light and assist it in moving in a particular direction. Multicellularity in Volvox probably evolved through increasingly complex adaptations that occurred after the shared common ancestor with Chlamydomonas. Now, scientists have noticed that there are proteins that are homologous in the unicellular Chlamydomonas and the multicellular Volvox. These are proteins that are involved in connecting the cells of Volvox. Because these proteins are so similar, it appears that slight modifications of genes, instead of the extensive origination of vastly different new genes, led to multicellularity. Let's examine this phylogeny that shows Chlamydomonas, Volvox, and some of their other close colonial relatives. Okay, the cells of Chlamydomonas are extremely similar in shape and structure to the colonial gonium. But the gonium possesses these adherent proteins to join to one another. Pandorina, a multicellular organism closely related to Volvox, has additional specializations. While Pandorina and Volvox have connected cells, they have an additional adaptation for keeping the cells moving together as a unit. They've evolved an outer cell wall that encapsulates the entire organism. And between cells, there's an extracellular matrix, or a substance that's fluid, but helps to stick the cells together. Now let's look at the origin of multicellular animals that occurred along a different lineage than the one containing Volvox. Scientists have determined that the group of colonial protists, called chonoflagellates, are the closest living relatives of animals. And the sponges are the most closely related animal group to the chonoflagellates. Now, the structure of the chonoflagellate cells and a particular cell type on sponges known as the collar cell shows amazing structural similarity, supporting this hypothesis. So just like the cells of the Volvox, animal cells have cell-to-cell junctions that allow cells to join and to communicate with one another. Cells need to be able to transmit messages from cells that take in sensory information to the cells that respond to this sensory information in order for a multicellular organism to function properly. The transition to multicellularity was probably the result of proteins being organized in different ways rather than the introduction of many new genes. When scientists look at DNA sequences and protein structure, it appears that the proteins that were present in chonoflagellates have been rearranged in a different order, allowing stronger connections between cells. So now that we understand that only slight modifications can lead to multicellularity, it's easy to see how multicellularity could have evolved so many different times.